Good morning. My name is Sheila Rao, and I'm here with you today to go through an overview of disorders of the proteome. So we are going to start first with learning some common terms. We'll explain how the environment, defined very broadly from both the micro to the macro level, can impact the life course of an individual. We'll build a repository of resources related to genomic conditions that one can use with clients, with families to explain genetic conditions. We'll also describe the historical context and the present day role of structural racism and its impact on the healthcare system, especially in relation to genetics, genomics, disorders of the proteome. There's a 2 to 3% chance for any couple to have a child with a genetic disorder or intellectual disability. Over 33% of all pediatric hospital admissions are due to genetic causes. And so it's important to have a better understanding of what we mean by genetics and genetic conditions. So congenital is a term that means that a condition is present since birth. A genetic condition can be congenital, or it may be something that becomes diagnosed and shows signs and symptoms later on in life. So not every genetic condition is congenital. Moreover, not every genetic condition is inherited. So inherited is a word reserved specifically for when a parent passes a genetic trait or difference to its offspring or children. Genetics is a term that refers to the study of genes and their role in inheritance. Genomics, on the other hand, is a study of all of a person's genetic material, the interactions of that genetic material, the interactions of that material with the environment, and the subsequent phenotypic changes. So this is a much broader way of thinking about the genome and of how genetic material impacts a person over the course of a person's lifetime. Epigenetics is another term that I would like you to become familiar with, and it relates to all processes that regulate gene expression in a given cell, leading to its cellular phenotype. Genetics uses information from one or two genes to explain a disease or condition, whereas genomics examines all of the genetic information to determine biological markers predisposing an individual to disease. The proteome, on the other hand, is the protein complement expressed by the genome of a cell at a particular time in a particular space. And so a cell may have one genome, but several proteomes, and we'll explore how that happens a little bit more. Just so that we are on the same page, I'm just going to walk you very quickly through some basics of genetics, and I encourage you to look at the website, the Genetics Home Reference from the National Institutes of Health, if you need to go over this in more details. So all of the hereditary material that are in cells are known as DNA. So there are 4 base pairs, adenine, guanine, thymine, and cytosine, that join together to form this characteristic double helix that you see here. And this becomes even tightly packed and denser, and is packaged in what I like to think of as chapters of the book, chromosomes. And so say that you have 3 billion pairs of DNA packaged into maybe 23 chapters of chromosomes. 22 of these pairs are autosomes, so that means that they are the same between both males and females, but the 23rd pair of chromosomes are the sex chromosomes, and they're either XX for female or XY for biological male. And these chromosomes have a very characteristic appearance, and that's how we came up with the numbers for them. There's a tight center part known as the centromere, and depending on the size of each of these chromosomes, we've identified where on these chromosomes are certain areas of DNA known as genes. Humans have about 20 to 25,000 genes. And so these are specific areas of DNA that are known to code for specific proteins. Some of these proteins have implications for diseases. There's also a lot of DNA that is not necessarily coding for protein. And we theorize that a lot of this DNA is involved with the control of the expression of genes from these different chromosomes, and may also impact whether or not genes are turned on or off in a cell at a certain point in time. So our DNA is extremely similar across organisms. On this slide, we have zebrafish, mice, chimpanzees, and dogs. And across these different species, we share about 70 to 82% of our DNA with zebrafish. We are hitting the 90s with the rest of the organisms on this slide. And so they're often used as models for genetic conditions because a lot of the sequence of our DNA is very similar across these different animal models. But the differences can occur because genes are getting turned on and off at different points after conception. There may be cues in the maternal fetal environment that cause these genes to get turned on and off. As I mentioned before, there are non-coding parts of DNA that are not involving genes that actually do decide whether or not a gene gets expressed, and single genes can code for multiple proteins based on whether or not they are being spliced differently. So if we were assembled together in a room and taking into consideration that you are not sitting next to a family member, you actually share more than 99.9% of your DNA sequence with an individual sitting next to you. We have very few differences in our DNA sequence. The average human gene will have maybe 1 to 3 base pairs different from one person to the next. And so for the most part, this sequence is preserved across human beings. Differences across species, however, are likely coming from the sophisticated control of genes that we tend to share. But how do genes change over a lifetime? They change because of epigenetics. And so those processes that are governing gene expression that may involve environments both within a cell and outside of the cell are what change the course of expression of genes, as well as the proteins that are turning on and off genes at different points in time. If you want more information, I encourage you to look at these different websites as well as YouTube videos. There are 3 different systems that we've known for epigenetic changes. Some of these include methylation of DNA, and so when parts of DNA get methylated, they do not get expressed into proteins. There's also modification of the way that chromosomes are packed onto histones. And so at different points in time, different parts of your DNA will be unraveled to be transcribed and translated into genes or into proteins. And those modifications of the histone can take place based on environmental cues outside of the cell. There's also non-coding RNA that can actually silence genes. And so when this RNA gets turned on by an environmental stressor, they may cause a gene to not be expressed, and that can have implications for the development of certain conditions. And we've seen this especially in the context of cancer. Therefore, we really want you to think very broadly about what is the environment of a cell. So on a micro level, there are chemical signals happening within a cell from 1 nanosecond to the next that are sensed by the cell and cause implications for how genes get expressed. On a larger level, outside of the cell, there are chemical signals that are taking place that are also changing the way that your DNA is being transcribed and translated, and these expression of proteins in the proteome are being literally impacted by things that are happening outside of the cell. And so this is the way that your lived experience gets encoded into the way that your DNA expresses your proteins. And so that's why my hypothesis to you is that possibly a lot of the conditions that we can't trace a very clear genetic cause to may have a proteomic level etiology. So our cells do send signals that are indicating alterations in these microscopic worlds. And our cells also trigger a cascade of genetic changes as a consequence. And these changes have different effects in the life course of a person. Again, if you want to take a look in more detail at some examples, these are great animated videos of gene silencing via methylation. There's also gene silencing happening at the chromosome level. So when we inherit DNA, I brought up the point that we have 23 pairs. So we inherit one set from one parent and the other set from the other parent. So only half of our DNA is often turned on at one point in time. And there can be different implications if the maternal copy is turned on versus the paternal copy for different genes. And so some genes will be effectively silenced, even though you have two copies, you're only using one. And that's what this illustration shows. There's a lot that we do not know about these epigenetic changes occurring over a lifetime. I did bring up that sometimes gene silencing can lead later on in the life course of a person to conditions like cancer. So there are some known changes that silence tumor repair genes, and that can lead to cancer. Another thing that you have to think about, though, is that there are some known conditions that we do have the tools to identify. So when we think about the types of genetic disorders, while epigenetics plays a huge role in how we experience different diseases over our lifetimes, we often turn first to looking at these sorts of types of genetic disorders in the tests that we use. So there are a very specific number of chromosomes, so we have 23 pairs, but we can have aneuploidies, trisomies, and monosomies that can have implications for conditions. And we can also have translocations of genes, we can have deletions, duplications of chromosomes, and mosaicism. And these are all sorts of different disorders that can be identified with different tests. We can also have disorders in the way that genes are expressed, and they are often having a very classical pattern of inheritance. And I'll show you some more slides related to some of these sorts of patterns of inheritance that are sort of our traditional understanding of genetic diseases. There's also mitochondrial disorders. All of our DNA for our mitochondria are inherited from our mothers. And sometimes that can have implications too for the passage of particular conditions. So we have genetic tests to look at the number of chromosomes, and that's the karyotype. With the microarray, we can look and see if chromosomes are the expected size. And so we might identify when there are duplications and microdeletions. However, if we need more specific ideas of the entire sequence of our DNA, that's actual genome sequencing. Other tests that we sometimes use, especially if we're particularly concerned about the expression of certain conditions that will change the way our genes are expressed, we can look at the protein expression, looking at things like plasma amino acids, urine organic acids. And we can also look at conditions with next generation sequencing, where we sequence out particular areas of the chromosomes where we suspect that there may be alterations in the gene. So here is a picture of the karyotype of an individual with trisomy 21. And over here you can see that there are 3 copies for chromosome 21 rather than 2. And this individual is an XY. So you see that there's a Y and there's an X, not 2 X's. And so therefore this is a male with trisomy 21. When you think about chromosomal epidemiology, it's rare to have a chromosomal number difference to begin with, because a lot of times these chromosome abnormalities have such broad implications on the organ systems of a person that a fetus cannot survive with these chromosomal differences. And so 50% of human conceptions end in spontaneous abortions, and the most common chromosomal abnormalities that end up as miscarriages are trisomies, triploidies, or monosomies. And there's a very high prevalence of chromosome abnormalities among spontaneous miscarriages or spontaneous abortions. Whereas the incidence among liveborns is, again, exceedingly rare, 6.2 in 1,000. So the most frequent chromosomal abnormality compatible with life is Down syndrome, otherwise known as trisomy 21, with 1 in 733 live births, roughly, reported by the CDC. When you think about genetic causes for different conditions, if you are looking just at the sequence of DNA, that's a very limited approach to how to identify different causes for different conditions. I want you to think again about the environment of the cell and the environment outside of the cell. And we have impacts outside of our cells with the way that these chemical signals are interpreted by our cells. So in the situations of large-scale population migration, some of our experiences in human history of migration and what is known as genetic isolation, you might see a change in the frequency of certain genes expressed in a population of people where over generations there is less migration of new populations of people to that place. However, I do want to make it very clear that there isn't a genetic basis for race. Race is a social political construct. There are commonalities of experiences in human history, whether you think of it as experiences over time of multiple populations, being exposed to the same temperature, latitude conditions. The melanin content of skin is very tightly connected to these sorts of conditions. And again, these are also absorbed and experienced by our cells over time. So vitamin D requirements can differ in different populations. There's also other experiences of human history, such as forced migration, systematic oppression, and structural institutional racism that do have implications as well for the proteome of our cells. So migration patterns can lead to visible differences, but not at the genetic level. So what I'm thinking about here is that, like, again, our sequences may be 99.9% the same. However, the proteomic expression of these sequences may be different based on our different lived experiences. And again, across all human beings, we have about 20 to 25,000 genes. So less than 1% of our genes are slightly different among people. And modern humans have only been around for about 150,000 years. So human genetic differences do not define race. However, there can be differences between different populations. And here you see a parent population of marbles that are yellow and blue. And this bottleneck effect is basically representing a specific environmental experience. And so perhaps a natural disaster or migration. And as a consequence, you have some significant reduction in the surviving population of marbles. And in subsequent generations, there are fewer yellow marbles because the survivors of whatever that natural disaster were only blue marbles. And so in the subsequent generations, you have a different frequency of yellow marbles compared to the parent population. Across the world, we have seen that, again, talking about temperature and latitude, the equator and countries that have equatorial boundaries tend to have a very high prevalence of malaria. So on the left-hand slide, you see in green the highly endemic countries where malaria has high prevalence. On the right, you see the prevalence of sickle cell factor. And this genetic factor in carriers gives individuals a slight selective advantage when it comes to having malaria. So having malaria in individuals who are sickle cell carriers is a frequency of this particular allele that has been preserved over time. Another thing that I wanted to bring up too is that the environment of the cell can also be impacted by other environmental stressors. So here on the left, we see a slide of annual infant mortality rates, and this is present on the Los Angeles County Department of Public Health website. And so individuals of African American ancestry have 3 times the rate of infant mortality, and this is just in these recent years. And one of the theories as to why is this happening is that there are environmental stressors that have been structurally set up by institutions that have continued to bring in racism, that have caused biological consequences as far as infant mortality rates in these individuals. And so obesity, undernutrition, stress, high altitude are causing maternal signals that are changing fetal growth and fetal programming. We have some tools for identification of genetic disorders, whether they're newborn screenings, looking for specific exam findings in individuals over time. We also have the capacity to look at patterns of inheritance in families. And so it's very easy with single gene disorders that have classic Mendelian patterns. So one of the patterns of inheritance that is fairly easier to detect is autosomal recessive. And so here you have a single gene that it tends to cause 25% of the offspring in a family of offspring born to individuals who are carriers will be affected, and 75% are unaffected. Whereas autosomal dominant inheritance is completely different, where you have about 50% of the individuals affected. There's also X-linked conditions with very characteristic patterns of inheritance where males are more often affected than females. And if you want to take a look at these patterns in more detail, you can definitely refer back to the NIH primer. When it comes to treatment of genetic disorders, sometimes diet and medication may reduce accumulation of toxic metabolites. Sometimes we turn to transplantation in order to try to alleviate conditions such as severe combined immune deficiency. Sometimes there isn't a treatment, there's just mitigation of secondary effects of the condition. Typically, we look for reasons to involve consultants for genetic testing when we're seeing evidence of failure to thrive in children. And this is just a clinical description, it's not a diagnosis in and of itself. Developmental delays, acquisition of milestones that are delayed, or complete loss of milestones. Sometimes there can be specific conditions that will prompt genetic referral, including autism spectrum disorder and epilepsy, as well as situations where there has been a known stressor in the maternal fetal environment. And our teams of multiple disciplines are usually assembled in the setting where we're treating conditions of genetic origin, such as in the CCS centers that you may be familiar with here in our hospital. Other specialty clinics can also have geneticists and genetic counselors as members of their multidisciplinary team. Beyond pediatric genetics, there's definitely genetic testing being done at the preconception level, prenatal testing being done nowadays, and also there's other sorts of testing, such as testing with websites like 23andMe and Ancestry.com. And these are not necessarily tests that are being done with the same level of rigor that perhaps tests that are being sent out specifically looking for things like the breast cancer gene, or panels of genes looking for specific chromosome locations for genes that are known to cause seizure disorder. And I encourage you to really think about the ramifications of testing for these different sorts of conditions, especially when we are testing individuals over the course of their lifetime for conditions that may not be occurring in the pediatric realm of their lifetime. And so, is there harm that could be potentially resulting from testing for a disease that won't be affecting an individual until their fourth or fifth decade of life? And also, what are the implications for family members of an individual once genetic testing has been done? Um, and these are all sorts of different quandaries that we have to confront and think it through. And we're going to be practicing that more with our workshop in the next subsequent weeks. And so overall, I want you to think of it this way, that like we have one genome that is very similar from one individual to next, but it can express the proteomes of several different lifetimes. And our lived experience is being encoded into those proteomes that are being expressed, both on a macro level and on a micro level. So racism has biological consequences. Temperature, latitude, migration, these all have biological consequences and can change us from the molecular level to the organismal level, especially in our risk of developing particular conditions over the course of our lifetimes and the lifetimes of our children. And so I do encourage you to question, what are the tools that are being used for science and are being deployed in the name of science? Who is using them? How are they being used? Who has access to them? Why they are being used? These are all some of the important policy issues that are currently confronting us as we are getting this wealth of information from genetic testing, genomic testing, and in the future, proteomic testing. And so again, here are some other resources at fingertips, but I do have additional resources for those interested in deeper dives. And remember, each individual in a family has a different lived experience of their proteomes. And so these two children are siblings. The little boy on the right has Russell-Silver syndrome, and he has a very different lived experience of the same, almost the same genetic information that both of them have inherited from their parents. But at the same time, they are very similar in so many other respects. Thank you very much.